my time doesn't enough for doing the chromatography and extraction, isolating only the compounds by MALDI-TOF and everything. So that's why I'm not conducting this part of the characterization and standardization. So, but in the future, like inshallah, if I continue with my PhD, so this part with the thing that you mentioned, so it's gonna go under the characterization with the standardization by the chromatography.
Okay, said well. Okay, you have a good explanation. Sana, one thing I want to ask, you notice or not, how many essay have you done total in all these experiment? How you plan that?
Sorry, doctor, like just...
How many essay, this kind of beta
okay.
glucan essay, how many essay actually? Do you count it actually?
Yes, doctor, like quite a lot of the essay like going on, it's a calorimetric essay. So first of all, like I will spend 3 months of my, inshallah, 3 months, 2 to 3 months until like done with my extraction characterization. So that's why like I need a lot of time to do all the calorimetric assay. I have a lot.
Yeah, you got this also. I think this one, because I'm not saying to look down on you, because as you mentioned, you keep mentioning you are mixed mode student, so you don't have really a lot of time to do this kind of thing. So you might don't want to too ambitious. Yes, you can finish, it's good thing, but too many essay also inside there. You notice also, right? Is some things cannot finish and look not good on your. This is proposal only, okay? I think you will see during the progress one. After that, then you can identify what you can achieve, what you cannot achieve, all this kind of thing. But now I think too much, but seem okay also lah, because I would think some of the essay is simply direct also essay, DPPH, something essay is not difficult essay also. Only on the macrophage and cell culture one. That one, have you experienced before using the cell culture before?
Sorry, doctor.
Have you experienced dealing with cell culture before?
Honestly, no. Only like in the first semester, we have a lab with Dr. Ong. So I have like basic and fundamental information and how the cell cultures can go. But I'm so like I'm so lucky. So I have my colleagues, the lab mate. So they are working on it. So I will observe their working and how they are dealing with the cells while like conducting my other parts of the research. So I have to like get some basic skills and how to deal with just like finishing my immunoassay part, cell-based assay part.
Okay, I think overall it's quite good, but I think I agree with Dr. Venu. You need to tie up your objective, your methodology, everything aligned together. So when you do a conclusion discussion, can align together. Just make it clear which one to which one. I think yours is quite clear. I think this slide also methodology quite clear also. Characterization which slide to do, but just to tidy up the things. Overall, is I think good proposal. I think good luck everything.
Thank you so much.
That's all.
Thank you so much.
Thank you so much, Sana, and thank you so much to examiners, Dr. Lai and Dr. Venu. So Sana, you can discuss with the supervisor, and I'm sure you're gonna do better. And good luck for your project. So I pass the session to Dr. Ong.
Yes, thank you, Dr. Shaida. Okay, let's continue. Next presenter will be Jing Tao Pan, student of Dr. Chung. The evaluators here, the examiners. May I know who are the examiners?
Dr. Eugene, and I can't remember the other one.
Dr. Eugene and Dr. Tasrik, he is already on the line. Kalau tak silap, wait. There. Dr. Tasrik and Dr. Eugene. Tasrik is from PJJ.
Yes.
Okay. Okay. So if everybody is here, then let's start. Go ahead, please.
Okay.
Eugene,
what?
Sorry, is Eugene here? I think Eugene is not here. You can better call.
Oh yeah, Eugene hilang pergi mana?
Oh, Eugene is not here. Okay.
Yeah, Eugene is not here.
Who is going to call him? Am I going to call him or?
Let me message him.
Okay, Dr. Chong. Okay, thank you.
Okay, so morning everyone. My name is Jin Tao Pan and my supervisor.
Wait. Sorry, Jin Tao. Dr. Eugene is not here yet. So we have to
We
wait.
should wait here.
Mm,
Oh, okay.
all right. All right. Dr. Eugene is here now. Dr. Eugene, okay, you are ready, yeah? All right. Jin Tao, you may start now.
Okay. Hi, morning everyone. My name is Jin Tao Pan. My supervisor is Dr. Chong. And my presentation topic is pentapeptide identification for the inhibition between cogranule BS189 proteins by in silico approaches. There are five parts of my presentation. First one, from 2016 to 2023, there are 370 new drugs were approved by FDA according their chemical structure. The number of peptides and oligos occupied 12.7% of new drugs. Despite the rise of small molecular drugs in 7 years, peptides still occupy an important position. So, short peptides are close effect products with high purity and more bio-friendly as bicycle biology molecules. However, they have some limitation, such as short half-time and low oral bioavailability. And we have some response methods, such as terminal protection and using natural amino acids can solve this problem. For those reasons, we consider using peptides to target surface disease. The short half-life would provide the benefits of safety. For example, a popular anti-wrinkle string, Matrix 3000, contains 1 4-members peptide and 1 tripeptide. And peptide can also used in ocular disease, targeting the CNV, and one pentapeptide used to simulate one water channel protein for tears to release dry eyes. So the peptide drugs is important, but how can we design or screen the treatment peptides? There are 4 ways to build peptide libraries. First one is chemical methods. When putting new amino acids into reactor, the FMOC protection groups keep n side of amino acids cannot form peptide bonds with other amino acids. So the amino acids only form peptide bonds with n side peptide, which link with solid surface. So the T-bags and solid phase peptide synthesis both given a surface to peptides attaching and prolonging. And OBOC is for produce more huge and rounding library, because after they prolong the shown terms and they miss and split, then so you can get the huge and rendering library. The second one is the biology methods. Biology methods is to produce DNA library first, and then put them into phage for merging with phage's DNA coding coat protein. So we can easily use the so those coding protein contain our potential peptides. So we can easily use targeting receptor to select our targeting peptides. And then last one method is the virtual screening. It can is a reusable and convenient method. It can contain any library size, but still require experiment verification. So why targeting S100A9 amylose is meaningful? The S100A9 monomer looks like M shape when in homodimeric situation, including 4 helix and 3 hinge regions. And S100A9 easy assembly to amylose, not only self-assembling but also assembling with other members of S100 family. And S100A9 amylose cause lots of disease, such as retinal toxin, Alzheimer's disease, and cancers. So S100A9, which is abundant in giresun and can spontaneously form amino structure, is involved in retinal function and dysfunctions. And S100A9 amylose is the attachment core of A beta fibers for progressing of AD disease. They have different kinds of structures, can be hydrophobic tempering and coating or low-static interaction with A beta fibers. And this picture shows the S100A9 on the surface of the A beta fibers. So how to screen the treatment peptide targeting S100A9 amylose? There are 3 S100A9 inhibitors can block the amylose aggregation of S100A9. The ApoE is 299 amino acids, longer than the S100A9 homodimer, which is 228 amino acids. Therefore, it appropriate to be a bigger clip used to cap the S100A9 homodimer in solution and restrict its extension. And research of this one, the polyomentalities design the sections of S100A9 from LYS 50 to LYS 40 as have a high amino propensity. And they describe the S100A9 amylo connect like occurs between both ends of each S100A9 homodimer. You can see this picture. And it was mentioned that only OLA, a natural oxidation chemical in olive oil, has the ability to disagree S100A9 fibers on the basis of blocking the elongation of exciting S100A9 fibers. And OLA shows peptide stack with TRP-80A, hydrogen bonds with ARG-85 and GLU-52 of S100A9. The the interaction with S100A9, which include the line, it includes the high amino prospective section from LOS 50 to LOS 54. So the ability to disgrade may be the result of an interaction force with the area from ARG 85 to TRP 88. So we think this area from ARG 85 to ALA 89 and from LEU 49 to VLO 58 of S100A9 is active area for S100A9 aminos. So this research screened 100 billion peptide phage display library against CAR proteins. It implies that the peptide can form highly interaction with S100A9. It can detect a concentration of core protein around tenfold lower than the mean found in a string of healthy persons. So choose the peptide to be a new drug is available. And my purpose, hopefully one day after validating the pentapeptide tree ability, the pentapeptide drugs can load on contain lessons for long-term treatment to tackling the like the acute prevention with conjunctiva. And furthermore, therapeutic pentapeptide may be delivered into brain from eyes to treat the AD disease, such as they deliver the nerve growth factors. My research urged first to construct a 5-member peptide library for virtual screen. Second, to identify the pentapeptides like binding with S100A9. The last one is to elucidate the interaction between pentapeptides and S100A9. So it's my methodology. Some 3D structure of molecules will be downloaded from the protein data bank and ChemSpider database. And ODA will be my positive control to compare the binding ability of peptides. And produce the pentapeptide library by RDKit Python API. It can transfer the peptide sequence to PDB form files. And the OpenAble will be used in converting the PDB to PDBQT fonts, because the PDBQT is the special font used in Autodoc. So binding free energy and hydrogen bonds will be analyzed and then ranking the Autodoc results. So we will choose some potential pentapeptide, and those more detailed analysis will be used the AMBER 2080 to do molecular dynamic simulation to discover. We can take the RMS, DRMS, and other interaction analysis. This one is my flow chart of this study. So we build the pentapeptide library first, and at the same time found out the active sites and found out the bicycle pyramids. We use the docking, and then we use the peptide selection by docking tools, and then choose the potential pentapeptide, do the MD simulation, and then do some data analysis. The last one is the sys write-up and submission. So at the same time, the exploration codes for batch process to handle the huge data. The next is my Gantt chart. I will spend 2 months for peptide library inspection and 2 months for peptide selection by docking, and 2 months for MD simulation. At the same time, do some data analysis and sys write-up. The last slide is my reference. I think that's all. Thank you.
Okay, thank you, Jing Tao. Any questions from evaluators? Of course, maybe we start with Dr. Tashrik. Dr. Tashrik.
Hello, hi. Thank you, Jinto, for your presentation. All right, okay, I have a few questions regarding your project. Can you hear me?
Yeah, I can hear.
Okay, all right. Okay, so because this project, you use the pentapeptide, means 5 member of the peptides, okay, or protein. So why did you choose this pentapeptides for this study? What the advantages do they offer over other type of peptide or molecules? Why you using pentapeptide? Why not octapeptide? Why not tripeptide? Why you choose pentapeptide for your this current research?
Because those true peptide or two-member peptide, they are too short. And from this, from this article, they have the 18 members peptide. So they already show the peptide has a good ability to binding with our receptor. So but if we choose the triple peptide, I think it will not to give us two more binding ability to our receptor, and tripeptide library will have the 80,000 peptides. The four member peptides will be mass 20, so it will be 1,600,000 peptides. And about our pentapeptide, will including the 3,200,000 peptides. So if we want to run because we think the pentapeptide library will be suitable for our project. It's not be too short and will be not be too long.
Okay, I understand what you're answering. Okay, so actually for the practicality lah, in this research you're using pentapeptide because you want to screen more.
Yeah.
More, and then you can get vast or big library, and then you can use again this as 100 A8 or A9 lah.
Yeah.
Or test. Okay, all right. But because when I look on your methodology, okay, for this constructing of pentapeptide library establishment, okay, I cannot you not mention in this research methodology how many library that you're expecting to generate, to produce and everything. So I think and then also you just mentioned about you using the Python library RDKit. However, you're not specific what actually the steps that involve. Like maybe you need to submit the sequence, okay, and then the sequence, the RDKit will create some sort of 5 member of peptide, and then maybe it will based on whatever lah. So I think they are lacking of the information for 3.1 in your thesis lah, and in your proposal. Okay, so I think you need to look back on this section, okay, and then try to improve lah by providing more information about how you can use this RDKit to develop or establish the virtual library.
Yeah, I told before, because this RDKT is one Python API, so I can use this one, convert the sequence to pentapeptide library. To build the 3-dimensional structure of these peptide molecules.
Okay. Yeah. I think you know lah more. But but please mention it more clear lah in your proposal or on your thesis after this lah.
Yeah. Thank you.
I think maybe we can give back to Dr. Eugene first, and then Dr. Eugene, and then
no problem. Yeah. Okay. Dr. Eugene, would you like to
Yeah.
start? Yeah,
Okay.
thank you.
Yeah, thank you, Dr. Ong, Dr. Ashrik. Good morning, Jintao.
Morning.
So, yeah, looking at it from, because you mentioned that it has already been screened, right? There are some in the slide 18. You showed that other researchers have also shown, have identified some peptides. I think the next one, right? Slide 18. Yeah, so is this the exact same target that you are going to screen against?
Oh, no. It's just an example. I just want to show the peptide is available very can very strong binding with my receptor. Because this one is for to detect this peptide. So you can see the longer peptide is binding in the outside of this protein. So it's not my active site for void amyloid. You can see this from is from I don't show like because it's in the outside, but you can see this picture is in is between the two helix in the middle of that.
Okay.
Yeah. So they are different.
So that means that you will be docking on a specific targeted area and not the whole protein.
Yeah.
Okay. And this active site has been shown to be the interacting site between the two monomers.
Yeah, because those inhibitors was published before they just show the potential active sites only in this region, like I said before, from 85 to 88 and from 50 to 58. Yeah.
Oh, okay, thank you. So that means that if you are able to block this region, then the activity will be lost for these two proteins.
Yeah. But actually, in my view, I think this one the polyaromatic they say the active sites only two sides of the S100A9, but I think it's maybe wrong. They just broke the connection. So this reason why they cannot disaggregate the performant S100A9 fibers, but the OLA one can. So I think I want to find the peptide can binding in these two potential active sites, because ODA including the 85 to 88, and the other active sites the 52, but this one ultimately is only binding the 50 to 54.
Okay.
Yeah.
But in your study, you will not be using the S100A8, right? So you are only focused on A9?
Yeah. Because this example, they want to targeting the components between S100A9 and AA. But this inhibitor only targeting the S100A9 amylose, only S100A9, no AA.
Okay. So targeting A9 alone may lead to maybe some kind of therapeutic use later on, right?
What mean therapeutics?
So targeting A9 may in the future potentially reduce, for example, retinal toxicity. Is that correct?
Yeah.
Okay, yeah. So the other question I wanted to ask was about the pentapeptide, which you have answered as well. So back to Dr. Tashrik.
Okay, thank you, Dr. Eugene. Okay, I have one question. Can you discuss how you use Python in your research? Because from your flow, exploring code for batch processing. But in your proposal, you not mention about how you use the Python, and you also not suggested any codes that maybe you can use for this process. So can you explain how you use the code for batch processing using the Python? Generally lah.
How to say that? um, Actually, it's a simple things. You just need to write some circles. The big circles including some small circles, and then you can do like autocomplete.
Okay. So this Python you're using what platform? You're using Terminal in Linux or Ubuntu or what? Or
Anything can is based on this API can support some platform, because there are some API only can use in Linux, and some only can use Windows. But at this project, I think if I don't use the docking API only can use Linux. But other one I test before, they can use in the Windows. Is okay.
okay. So actually, I think better if you put in your proposal, you can specify the task that this automated Python script has been done lah. Because you not mentioned, because you need to process all the batch, all the data after you got from the library, virtual library, and then of course, because like example, after you got few billion, I don't know lah, few structure of pentapeptide, at least you need to have the Python code that maybe it can filtering what the criteria that you will choose for the next, for the docking. You will not dock everything from the virtual library, I think. So at least in your proposal, you need to show the step lah, the Python code that you will be use and everything lah. That's make it clear lah. Because now you just put in the flow chart. You mentioned that exploring code for the batch processing, but in your proposal, okay, I didn't found any section that this the code, this the step and everything. So I think that maybe you can include more lah in your.
I know you mean, but I just think if I show my code, it's difficult to.
Oh, okay. No. I just mentioned that maybe you can put like example, data processing.py. Okay, this code, and then you mentioned that this code will using for the process all the peptide that generate from the virtual, and then give based on this criteria for the further processing with Autodoc, Vino or Autodoc 4. Then we know lah that you have this type of Python script and everything, the step and everything. So that's my suggestion lah, in order for us to look more clear about your project lah.
Oh, yeah.
I
think in the next progress presentation, I can show you my strategy of the build the library.
Okay, all right. Because
Okay,
and
thank
then
you.
I think wait okay. Let's say if you finish everything and then you got the peptide that you potential peptide and everything is bind at the active site or maybe passive site, I don't know lah. So and then this can prevent from the aggregate of this S100A9. So actually, how will your research contribute to the field of peptide drug development? So in the future, what actually this data can provide to the scientific community? Any idea?
Sorry.
Okay, let's say you finish, you identify the RMSD, you identify the interaction residue and everything, and then it shows that this AMBER simulation, it can block the aggregation of this S100A9. So what actually is contribute to the drug development? What the future direction that?
Oh, yeah. I think that's why I choose this project, because I want to target the surface disease. So we just deliver to your eyes, and we can put in the drop and drop in your eyes. It's okay. So it's not too difficult for me to deliver the drug to my target disease. But actually, this S100A9 also have in some cancers lay in the middle of your body, in the deep side of your body. But in this case, I just want to deliver to solve some eyes disease first, and then maybe someone can have some deliver strategy and deliver my peptide into the deep of the body.
Okay, alright. So you need at least after this maybe the future direction there are researchers that can use this peptide conjugate with anything and then deliver it into maybe cancer cells and then see the effects directly lah. So I think that's the future direction lah. Okay. So also you need to mention lah for the verification or validation by the in vitro or experiment lab for future direction lah. Alright. Okay, I think that's all from me lah, Dr. Eugene and Dr. Ong.
Yeah, I have no further questions also, Dr. Ong. Thanks.
Okay, go ahead, please. Dr. Eugene, do you have any questions?
Yeah, I don't have any other questions.
Oh, you don't have any? Okay, then we stop here for Jing Tao Pan. So thank you very much.
Okay, thank you.
For that. And thank you, Dr. Tashri and Dr. Eugene, for your comments and suggestions. All right, now I pass to Dr. Chong.
Okay, thank you, Dr. Ong. So we shall proceed to the last presenter for this morning session, Song Jia Yi. Because I was not given the evaluator's name, may I know who's the evaluator for Song Jia Yi?
Dr. Eric and Dr. Khoo Boon Yin. Yeah.
Dr. Eric and Dr. Khoo. Okay, so Song Jia Yi, you may start your presentation now.
Okay. Good morning. I'm Song Jia Yi and my supervisor is Dr. Ong. My research project is elucidation on mechanism of antiproliferative properties of Camelina communis against cancer of rich cell lines. Camelina communis, also known as Asia dayflower, is an annual plant native to East and Southeast Asia, and there are a variety of active ingredients such as flavonoids and alkaloids. These ingredients have many pharmacological activities, such as anti-inflammatory and antibacterial, antioxidant, and antiviral. In traditional Chinese medicine, Camelina communis is widely used for clearing heat, detoxifying, and reducing swelling. Studies have shown that different species of Camelina have shown significant activities in pharmacological. This part will focus on the research of these plants in cancer. For example, the methanol extract of C. benihalasis has antiproliferative effect on leukemia-targeted cells and achieves G1S phase arrest by affecting K cycling. Additionally, it also has anti-cancer effect on oleic acids mouse model and shows cytotoxicity and apoptosis induction in human cancer cells, including MDMB-231. Methanol extracts from other Camelina plants also showed antiproliferative effects on oral cancer cells and are able to induce apoptosis and DNA damage. Although Camelina species plants have shown extensive potential in anti-cancer research, the specific research on Camelina communis is still limited. So the originality of this study lies in the fact that extracts from other Camelina species have shown antiproliferative effects on various cancer cell lines, and these plants have been widely used in traditional medicine, with proven pharmacological activities. The presence of these chemical compounds provide a theoretical basis for the potential of C. communis in cancer treatment. So this study is to explore the mechanism of communis inhibiting cancer cell growth and fill the gap of existing research. Best of both the research objective is to evaluate the anti-proliferative effect of C. communis and explore its mechanism of regulating proliferation in cancer-origin cell lines and non-cancer-origin cell lines. The research question is, does the extract of C. communis have anti-proliferative effects on cancer-origin cell lines? And by which mechanism does the extract of C. communis inhibit the proliferation of cancer-origin cell lines? Is the aqueous extract of C. communis cytotoxicity to non-cancer origin cell lines? Are there differences in anti-proliferative activity of C. communis among different types of cancer origin cell lines? And the experiment flowchart as follows. I will start from plant collection and identification, sample preparation, extraction, standardization, and the cytotoxicity assay. If the cytotoxicity assay meets my expectation outcome, then cell cycle detection and apoptosis detection will be carried out, and then we'll do the data analysis. In methodology, because water decoction is widely used in traditional Chinese medicine, and it is a relative mine extraction method, and consider. So I will, after plant growing, I will boil in the extraction for 30 minutes. And considering the impact of solvents on cytotoxicity, this experiment will use water as a solvent for boiling extraction. After extraction, standardization tests will be conducted to ensure the consistency. The cell lines will include the breast cancer cell lines, MDMB-231, and the MCF10, and non-cancer cells, MCF10A. A bottle of drug concentration will be diluted twice from 200 to 0.75 micrograms per milliliter, and the cell cytotoxicity detected after 24, 48, and 72 hours. If the MTT results meet the expectation, and the cell cycle and apoptosis detection will be carried out for the mechanism research. Because considering that other plants of Camelina have been proved to have the effects of cell cycle arrest and apoptosis, so I choose this test. About the cell cycle test, we use PI staining and flow cytometry to measure the disruption of cells at different cell cycle stages. And the caspase cause caspase protein family plays a key role in the process of apoptosis, and its increased activity is one of the hallmarks of apoptosis. So I will choose fluorescence assay to do the caspase assay. And best of all, my expected outcome is the aqueous extract of C. communis can inhibit the proliferation of cancer-origin cell lines, and the anti-proliferative effect of aqueous extract of C. communis may be achieved through inducing apoptosis or cell cycle arrest in cancer-origin cell lines. The extract of C. communis is non-toxic to non-cancer-origin cell lines. Milestone is I plan to in May obtain a crude extract and a standardized spectrum. In July, obtain cytotoxicity test results. August got cell cycle test results, and September got cell apoptosis results. October do the data analysis. And this is my Gantt chart. We use 1 month to do the plant collection, identification, extraction, and HPLC. After that, cytotoxicity assay and the cell cycle apoptosis each part will use 2 months to do it. If all goes well, I will finish my experiment in September, and October will do the data analysis. That's all my presentation. Thanks. Any question?
Question from evaluator. Maybe Dr. Khoo, do you have any question to ask Sung Jia-Yi?
Thank you, Prof. Chong. Jia-Yi, can I see your milestone again?
Okay.
Only 6 months.
Yes.
Are you sure?
If all goes well, 6 months should can.
6 months only. Yeah. You sure you have only 6 months, to complete everything?
Yes, cause I already got a cytotoxicity preliminary results and it's good.
Yeah, this is not a question, it's just to confirm with you the timeline, because 6 months, you may not able to finish everything. Okay. Can I comment on your proposal? Do you have the proposal with you?
Proposal review, this part literature review.
No. Do you have proposal with you now? Yeah, I know you put everything on the slide, but can you see the abstract? Just in brief. So your study aim to evaluate the effects of this plant on the cell cycle proliferations. But in terms of the write-up, I think you are not consistent, because when we go to the when you go to the objective, you see the objective in your slide, same thing. You use another statement to evaluate the proliferative effect and explore its mechanisms of regulating the proliferative effect in cancer origin cell line. I think this statement sound better. Okay.
Okay.
Yeah, so I think make it consistent, because you are not in the mechanistic study yet. You study you try to study the mechanism related to the proliferative effect, which are cell cycle and apoptosis only, right, as stated in your proposal and also the presentation just now. Okay. Right.
Okay.
So focus on this few things, proliferative effect. I'm not sure your MTT essay going to determine proliferative effect or just to assess the viability, cell viability. Maybe can you explain?
Explain.
Your MTT essay, you state you want to study the proliferative effect, but you use MTT to assess it. So your MTT essay, you did mention this is viability test. So whether this test determine the cell viability or cell proliferations, can you explain to us?
About MTT, I also said the control group and the blank group, and the water extraction dilution will start from 200 micrograms and value twice to 0.75, and each cells will plant 3 batch. Each cells will do 3 plays, and for 24, 48, and 70 hours, and then can get the results.
You are explaining the MTT essay, right?
Yes.
So this essay, listen properly again. This essay determines cell viability, or it will provide the cell proliferative effect to you.
It provides the cell availability to me.
It's okay. Maybe you can discuss with your supervisor later. Can I see your objectives in your slide? I think you have this specific objective in your slide. No, these are research questions.
Or this part objective.
This is objective. How about specific objectives?
Specific.
Maybe in your proposal, right? Okay.
Yes.
So in your proposal, you list down 4 specific objectives. First is to prepare aqueous extract. This is not objective, huh? This is
Oh.
methodology. Okay, so please remove it. And then the second one is to assess anti-proliferative effect find. And then the third one is to identify cell death pathway. Cell death pathway. Yeah, so cell death pathway. What cell death pathway do you mean? Cell cycle, apoptosis?
Yeah. I will use cell cycle and apoptosis to just.
So please be more specific. Just don't use some other words. Just focus on cell cycle and apoptosis.
Okay.
Okay. So remember cell cycle and apoptosis.
Okay.
And then one more thing in your proposal. Refer to your proposal again. Expected outcome in your proposal. This time you did mention the apoptosis, but you miss out the cell cycle. See your proposal. Expected outcomes.
Expected outcomes.
Page 14. Page 14. I will send the review proposal to you. The proposal I have reviewed, and I put my comments back to you. Okay, later.
Okay, thank you, doctor.
All right. One last question. In your proposal, page 15, you have planned for future work. What plan of future work you want to carry out? Because your current work has not started yet, right?
Yes, haven't started.
Yes.
But
So
500
is that necessary to include plan for future work? Page 15. 15. Yes. Plan for current work, I think. Plan for current work. That means your proposal. Because you don't even finish your current work, so how would you plan for your future work? Yeah. Okay. So maybe you can try to review your proposal and send that to the office later. Okay. That's all from me. Thank you very much.
Okay, thank you, Dr. Khoo. Question from Dr. Eric.
Thank you, Chairman, Chairpersons. Sonjay, okay.
Okay.
Overall, it's good. The proposal. Just want to know what the research the expected research outcome. I think is this common plant have been used for other research. What's your research gap or outcome? Want to fill the gap of research in other research? What you want to achieve for your research? Can you explain more?
Cause previous research is focused on the other plants of Camelina communis, but cancer research on this communis is not is limited, and there are just focus on the anti-inflammatory, antioxidant. Do not do the anti-cancer research.
One thing I think this slide also in expected outcome, I think some spelling mistake also. One thing is a cancer original cell line. What does it mean? What does it mean? Why didn't mention the specific name? Why is mean like cancer original cell line? Can you explain more?
This plant can have work to the human cancer cells, such as cause this proposal I choose 3 cells. One is non-cancer cells, and another 2 is to MDMB 231 and MCF7. So I think this all are 2 cancer cells.
I think it's better, yeah, that you spell out the cell line or you name the cell line rather than, because this is unclear because you put a cancer origin cell line or non-cancer origin, people cannot, I mean, the examiner or who want to ask the question, they don't know how to ask also. Because it's a very general statement. So it's better that you put a specific name. At least they can check, okay, whether this cell line have been checked before, maybe what FCS7 or other thing, what non-cancer cell line. It's better to name it that compared to this one non-cancer cell line. We don't know what's non-cancer cell line. At least you name it. Or you want to put a specific name there, after that you put it as non-cancer cell line, it's okay. At least we can check. Now we cannot check what's non-cancer cell line. It's too many also, right? Cancer original cell line, I saw too many cell line. I don't know what you want to compare also. Okay? I just now listened that you complete the cytotoxicity already, right? The first milestone? Can you go back to the... You said to Dr. Koo that you complete the Gantt chart, go to Gantt chart there. Yeah. You said you complete the cytotoxicity already.
Just go to preliminary results.
Oh, the preliminary results. So you complete the SPLC, you complete the plant extraction already?
Complete this part, this haven't complete.
Okay. So SPLC not complete yet, but cytotoxicity, you carry this part of the cytotoxicity study already lah.
Yeah.
Okay. Okay. So so far the progress is on time. But okay, I think overall I don't have questions. But I think just to same like Dr. Koo also, I think you make sure you have the research is in line with your objective also and expected outcome also. I say the objective also, you don't only put a general objective on the presentation. I think you should list out all the specific objective also.
Okay.
And as you know, the specific objective might be SMART. I think the SMART word, I think you must be measurable, all these kind of things. Quickly, I think research objective questions or this kind of thing, you should put objective and followed by specific objective, so people can follow the study. Okay, I think the cancer origin cell line, you should put it somewhere lah, and some replace it with a more specific name that we can compare. Let's say breast cancer cell line, you know, at least you put a breast cancer cell line, then we know what is it. You don't know cancer, I don't know which part you want to study as well. Okay, be more specific because you do final project, so it's not a very general thing unless you're not you cannot determine at this point lah which cancer cell line you want to do, then it's okay lah. If you determine which, let's say breast cancer cell line, colon cancer cell line, this kind of thing, then you stated out and the things, at least we can do a comparison. And this one, at this point lah, maybe you still not carry out the research, so still okay. But I think during the progress time, I think you should spell out, I think, things. Okay.
Okay, thank you, doctor.
Okay, how you want to go back to the methodology again? Next two slide. Next slide. Okay, methodology. Next. Okay. This one, number 7, slide number 7. Okay, this boiling, you use grinding, boiling, standard of 10 standardizations. Okay, at least your standardizations. C capsules. Okay. But here you got MTTSA, you got MDA, MCF, NCF, all this kind is a cancer cell line. This is what? What is it?
What? Sorry?
MDA, MCF7, MCF10A. This is cell line you use, right?
Yes.
So
MDMA 231 is TNBC cells and MCF7 is ER cells. And MCF10A is breast personal cells.
So you name the cell already. So I think better when you put on objective, it's better you change to the name like breast cancer cell line, this kind of things. Okay, that's all for me. Chairperson, thank you.
Okay, thank you, Dr. Eric, for your comments, and thank you again, Dr. Koo, for the comments. So we shall now end the morning session for the presentation. The evening one, we will start at 2 o'clock, and the session will be chaired by Dr. Sasi. So with that, thank you for your attention. See you in the afternoon.
Kevin, merci.
Next is to investigate the anti-inflammatory activity by measuring the key inflammatory markers like TNF-alpha, interleukin-6, interleukin-8, interleukin-1b in MCF7 cells. Next, the research methodology. It involves identifying the antioxidant activity, anti-proliferative activity, anti-inflammatory activity, and sample profiling of commercially available lavender. So for antioxidant activity, I'm gonna follow DPPH assay. So first I'm gonna prepare the sample standard and the DPPH solution. I'm gonna mix on a 96 well plate and incubate for 30 minutes in the dark and read the absorbance after 30 minutes at 517 nm, and then gonna make a graph for radical scavenging capacity and effective concentration 50%. Next, for anti-proliferative activity, first I'm going to start with cell seeding in a 96 well plate and then taking the cells with different concentration of the lavender essential oil and then incubating it for 24 hours, 48 hours, and 72 hours, and then adding MTT and 5 mg per ml in phosphate sulfur saline, and then incubating it for 4 hours in 37% 37 degrees Celsius and 5% CO2. After that incubation, I'm going to remove the media and add 100 microliter of acidified propanol, isopropanol, and incubate for 5 minutes, and then add autoclaved deionized distilled water and extreat absorbance at 570 nm, and form the dose response curve for this assay. Next, for anti-inflammatory, first gonna harvest and count the cells and prepare appropriate cell concentration for the seeding in the seed cells in a 6 well plate, and next add various concentration of lavender essential oil to the plate and then incubate it. Next, I'm gonna centrifuge the cells and take the supernatant and run it with ELISA kits to quantify inflammatory mediators in the supernatants, and then identify the significance through SPSS software. Next, for sample profiling, I'm gonna run for various tests for different chemicals. So test for tannins, I'm gonna add 0.1% FeCl3 and gonna look for blue-black coloration or brownish-green coloration. For phlebotanins, I'm gonna add 1% aqueous HCl and I'm gonna look for red precipitates. For saponins, I'm gonna just add distilled water and shake vigorously for 5 minutes and look for honeycomb froth. For flavonoids, I'm going to add 1 ml diluted NH3 and 1 ml concentrated H2SO4 and look for a yellow coloration in the solution. Next, for terpenoids and steroids, I'm going to add 1 ml chloroform and 1 ml H2SO4 and see if there is a distinct reddish brown coloration in the interface. Next, for cardiac glycosides, I'm going to add 1 ml of glacial acetic acid and 1 drop of FeCl3, and also add 0.5 ml of H2SO4, and we'll look for a brown ring at the middle surface and a violet ring below brown and greenish rings. Next, for anthraquinones, I'm going to add chloroform and a 10% ammonia solution and look for a red, violet, or pink coloration. Next, for reducing sugars, I'm going to add 1 ml of each of filling solutions and going to look for brick red precipitates in the solutions. So the expected outcomes of the study is to identify the chemical compositions. Next, demonstrate free radical scavenging capacity of the commercially available lavender essential oil, and next, investigation of the anti-inflammatory effects and assessing the anti-proliferatory effects in MCF7 cells. Next is a summary for work in progress. Investigating the biological activities of Lavandula angustifolia essential oil, evaluate antioxidant, anti-inflammatory, anti-proliferatory properties. Next, so overall, this study aims to provide valuable insights for the therapeutic potential of the commercially available lavender essential oil. This is the gun chart So I'm gonna take 1.5 months for antioxidant assay and 2 months for cell viability assay and 2 weeks for phytochemical assay and 2 months for anti-inflammatory and statistical analysis is progressive. So at the end of every experiment, I'm gonna do the statistical analysis for every assay. These are the references. Thank you.
Okay, thank you, Sultana, for your nice presentation. So now I'd like to open for Q&A session. Maybe we can start with Prof. Kubunin.
Thank you, Prof. Sase. Sultana, can I see your expected outcome again?
Yeah, sure.
Yeah, this is just my comments. Not really a mistake. So you have 4 expected outcomes. Chemical compositions which will be achieved by sample profiling. Is that correct?
Yeah, for characterizing the chemical
Yeah,
composition.
and then the free radical scavenging is by antioxidant, and then anti-inflammatory, you have your assays, right? Anti-proliferative also you have the assay. So basically your study focus on 4 things. Sample profiling, antioxidant, anti-inflammatory, and anti-proliferations. So can you go back to your specific objective? So 1, 2, 3. So 1, 2, 3. Where is the sample profiling? Is that it is not part of the objective of your study?
It's like it's gonna add up.
Or it fall under which? Under antioxidants?
No. It's a separate thing, but it's a research kind of objective. It's not the main objective of the study. So
it is not the main objective. Is it the one of the specific objective? One of the objective? No. You may consider it. To me, you have 4 things lah. So your research questions also you should have 4 questions. Specific objective also 4. And then what else? Even in the abstract, if you have your proposal with you, your proposal also you did mention 4 things: sample profiling, antioxidant capacity, anti-inflammatory effect, and also the cytotoxicity through cytotoxicity assays. 4 things. Okay, so try to make it consistent. Right?
Yeah. Okay. Okay, doctor.
Yes. What else? All right. You have your proposal with you? Do you have your proposal with you?
Yeah.
Your proposal with you. Maybe I can also comment on the backgrounds. I think you don't have to put so much information on the breast cancer like BRCA1, BRCA2 because you are not really determined these genes right in your study. See page 4. So or you really want to include these informations in your proposal. Maybe you can explain or answer how your essential oils are able to reduce invasive and non-invasive cancers, breast cancer. Maybe you can explain it, but not just saying it because this is some things sound like not really related to your study.
Okay, doctor. I got it.
Got it. So can you see the research questions? Page 6. The research questions 4. Does the chemical compositions as a therapeutic remedy in treating breast cancer? Is this your research questions? At the end of your study, you are able to answer it. See whether this essential oil, is that correct? Can be used as a therapeutic remedy. I think the word therapeutic is too big. Too big. Okay.
Okay, doctor.
So, may I ask, what do you know about the current treatments for breast cancer? Do you think you should also have a positive control for your study? Sultana?
Yeah.
Okay, currently, what methods, what strategies doctor use to treat breast cancer patients?
Yeah, there is surgical removal of the tumors and also chemotherapy. I don't remember the exact drugs that are used nowadays for the different mutations of the cancer.
So, the essential oils
yeah, it can be a supplement with the
but not the therapeutic agents.
Okay, so I should just write it as indicate
It may
potential.
be used to
in-
prevent breast cancers, to prevent chances of getting breast cancer, but I don't think it can be used as a therapeutic agents or remedy in treating breast cancer. Can we go to methods? I have a questions. Methods. Have you started your experiment?
Yeah, I'm currently working on antioxidant assay.
Can I see your Gantt chart?
Okay.
So where are you now?
I'm in antioxidant assay, so I'm still like precising the antioxidant assay. Like I did it before. Now I'm gonna narrow it down more.
You have completed the antioxidant assay.
No, I'm still working on it.
Yeah.
Yeah, so mid-June I will be able to complete it, hopefully.
Okay. Can I ask a questions for your survey essay? Can you see page 14? 14, no? Can your I'm not sure whether this is correct or not, because the word oil, right? Oil, yeah? Oil usually won't mix with water, right? So quite curious, can your essential oils treat the cancer cells maintaining with cell culture medium?
That I haven't looked into.
Because even you mix perfume, right? If you have perfume, you can try. Even you mix perfume with water, the perfume always the perfume will not mix with water. So I'm curious whether or you have a specific way to treat your cells. But you did not specify it in your proposal. So I wonder how your essential oil able to mix with the cell culture medium and thereby give some effects to your cancer cells, MCF7.
For the cell culture medium, I'm going to use as ethanol. So if there are like studies which show that
you will mix the essential oil with ethanol.
Yeah.
Oh, it may be possible. I don't know. I never tried. I don't know. Okay. Right.
Okay.
So I think that's all from me. Thank you.
Thank you, Doctor.
Thank you, Prof.
Yeah, thank you.
Sultana.
We move to Dr. Eric. Any question?
Okay, thank you. Thank you, Sultana. I think interesting research. So I think I share the main concern, Sam Dr. Khoo also, because here you put the title is essential oil. You know, it's not the crude extract from lavender or this kind of thing. So how many percentage of the oil or this kind of thing can we I'm not sure because hardly can mix the things. I'm not sure because I think can you go back to the Richard review on the slide? Yeah, it's all this kind of Richard review, right? Is are they using essential oil or they use the crude extract for lavender? This one, I say all this farmy everything.
So some of the
Some
researchers they
extract, you
use
see, right?
extract, some
Extract. All this they put extract, you know. Found is the compound in the oil. Okay, the number 2 one is yeah. So I'm not sure because that's the problem you're gonna face with because this is proposal, right? So that's the problem. I'm not sure maybe like Dr. Khoo said you got special method. Let's say you mix with ethanol or this kind of thing. Not sure because you cannot 100% guarantee that the essential oil, the compound inside the essential oil gonna go inside the cell and give the effect. Whether it's totally give effect. If say no effect, whether because the compound itself cannot go inside the cell, we don't know. That's why I think important thing you want to highlight actually the positive or negative control of the study. I think maybe you use essential oil or other thing that similarly property that you can conclude the study. If not, if you go in, yes, a good thing lah. They got effect. But if really it cannot go in, then no negative, no result. Whether the result show that is because of the essential oil or is because the other thing property because it just cannot go in. You understand my point, Stana?
Oh yeah, yes, Doctor.
I think this the tricky thing lah. I'm not sure because I don't know. I don't know how many percent. I don't know how many if you mix with ethanol other solvent, how many percent we go in also we don't know. I think better that you keep the positive control, negative control as a study inside there. So that will be helpful lah to explain. If not, you cannot explain the thing, then that's the terrible thing lah to when you want to defend your thesis. My point there also. Okay. When you go back to the methodology. Okay, the floor one, which one the floor? All the floor together now. This
Sorry?
one, okay, this is okay. So this show antioxidant, anti-inflammation, anti-inflammatory. Does this, I think the results show in the literature review already? No? Some part is show in the literature review already. So what's make you different? Why you want to repeat the experiment? Or what you want to show compared to the result in the literature review? Can you explain more?
So the previous results are with the extract, lavender extract, and here I'm gonna work with the commercially available lavender essential oil. So it's not extract. I'm not dealing with extract.
Yeah, but some the number 2 one for me is show the crude oil, right? You go back to oil, you see, for me is 2, right? I think found 60 compound in the oil, right? You see, this is oil for me.
Yeah, the cell lines are different here on my
so
cell
I
lines.
think you need to differentiate a bit when you do the explanations. What's so different? Because it's okay, I think, for mixed mode master, just to show a different parameter, different cell line. It's good also because the oil one they use have G2 and A50549 cell line, right? You use MCF7, right? There's a breast cancer also quite different also. So
Yeah.
that's a different thing also. At least because why I raise this, because at least you can report, publish the thing. It's good to publish the result. And I think it's at least I think not the student face in the mixed mode, actually they don't have publication. When they don't have publication, you don't publish the result, you don't have publication. When you want to apply for PhD or scholarship, that will make your application quite not very in positive got merit. So it's good to you publish, the result, something, or maybe the review paper also. At least it got some publication. If not, it will make your application for job or PhD a little bit more difficult actually. Especially, it's easy to get a PhD, but difficult to get a scholarship. That's the problem. So I hope you will learn. That's why I want you to publish the result. At least you quantify positive, negative control. Whether I think important thing actually. Okay, understand? You understand my point?
Yes, doctor.
Yeah. Okay, go back to expected outcome. Last one. Okay, characterize the chemical composition. This one you use SPSC, right? This one, right? Which one?
For chemical composition, I'm gonna do the sample profiling, like
profiling.
the
Oh,
way
this
you
is
did.
profiling. So are you gonna use SPSC or SAMS for this one? No. Just profiling only.
No. For anti-inflammatory, I'm gonna use SPSS. I'm gonna use ANOVA.
No. You mean LCMS or SPOC for the characterization, because you just use the
SPSS.
chemical test only, right? Whether it's compound, acid or not acid, right? Inside the
Yeah.
So I'm not sure. This one you can discuss because all will involve additional research fees also. So I think not sure lah, because like I say some other literature, I mean other paper also they will list out the chemical compound also, whether it acid or not. Let's go compare. One thing is my concern actually, the essential oil you get, where you get the stock? You mean from the counter of the cosmetic or this kind of thing, or somewhere, or it mean you have a selection of the essential oil? How you get the stock? May I know?
Got it from the manufacturer.
Manufacturer, somebody provide the lavender essential oil.
Yeah.
I mean the standard lah. I mean come from one sources lah. I mean at least one brand or this kind of thing. I'm not sure. This one you have to check because when you present in the progress, at least you have to mention whether it's provided by company or the brand or this kind of thing. At least you have to mention, so people next time they want to repeat the experiment, they will get the same thing. Because sometimes why they want to get a repeat experiment when they get a positive result. Let's say you anti-cancer or this kind of thing lah. They want to repeat, then they will want to know the company who provide the thing. At least I think you've read. Okay, the one thing is why you want to do the free radical scavenging activity? Why is this important for your research?
It's because the cancer cells will have a lot of oxidative stress, so it would add up to the study that the oil is having a antioxidant capacity.
So this is your theory lah. I mean, using the essential oil probably gonna reduce the free radicals inside the elements. Okay, I think you need to carry out the experiment to prove it actually. Whether it's got a positive or negative, I think you need to report it. Okay. Overall, it's good, suitable for mixed mode. I think main concern is whether the oil is mixed with how you gonna mix the thing lah. Maybe got formulations. I'm not sure lah. Discuss with supervisor because of interesting project actually. I hope you good luck. That's all for me, Sultana. Thank you.
Thank you, doctor.
Okay, thank you, Dr. Eric. So Sultana, please take notes about the concern raised by both examiners. So now I pass the platform to Dr. Wong to chair.
Yeah, thank you, Dr. Sasi.
Thank you. Yeah, thank you.
All right. Let's go to next person. Tapa Maga Puspa. Are you ready? Hello.
Scary. Oh.
Tapa says, wait. Tapa, are you ready?
Hello.
Yes. So okay.
Is my voice audible?
All right. Let's go to Tapa now. Tapa is a student of Prof. Madhya Dr. Sasi. So please go ahead if you are ready. May I know who are the evaluators for Tapa?
Dr. Chu Ailan and Dr. Saida.
Okay. So both of them are here, right? Dr. Chu Ailan is here. Also, is here.
Yes, yes.
Okay. All right. So then we can start. Tapa, please go ahead.
Can you see my slide?
Yeah, we can see your slide.
Can you change to presentation mode?
Yeah, change to presentation mode on your right hand lower corner. Yes. Okay, please go ahead.
Thank you, Doctor. Good afternoon, everyone. I am Puspa Thapa, and today I'm gonna present on the evaluation of antimicrobial activity of medicinal plant-based green carbon dots against pathogenic microbes. My supervisor is Dr. Sasidharan Srinivasan. These are the contents. Introduction. Sorry about that. Introduction, carbon dots. Carbon dots are fluorescent nanoparticles, which is the carbon-based nanomaterials, which were discovered in 2004 by a group of researchers accidentally during the purification of single-wall carbon nanodots. Carbon dots are nano amorphous and quasi-spherical in shape. Quasi-spherical, it means the shape generally is spherical but has slight irregularities at its end, as you can see here, surface deviation, which doesn't make it a perfect sphere. They are zero-dimensional, which means that they are like a dot point-like structure with no measurable size in any direction. And carbon dot, they also contain many functional groups such as amine, carboxyl, hydroxyl, etc. And the core of carbon dot is composed of sp2 and sp3, which is responsible for optic properties. These amine groups or functional groups are responsible for one of the very important property of carbon dot, which is water soluble. This groups makes it possible. And the size of carbon dot is 10 nanometer or less. There are various advantages or properties of carbon dots, such as biocompatibility, environmental friendly, cost-effective, multifunctional functionality, high surface area, and most importantly, like antimicrobial properties, and it is easy to synthesize. mechanism of action of carbon dots. Carbon dots have recently emerged as an promising candidates for various biomedical applications, including antimicrobial agents. At the moment, the exact mechanism of antimicrobial activity of carbon dot is not yet known. However, several hypotheses or ideas have been proposed. So carbon dots can act in the presence of light and also in the absence of light. So in the absence of light, it disturbs physiological metabolism of microbes, inhibits biofilm formation, and also like when it can bind to DNA and RNA as it is really small in size. So as carbon dots are very small, they can easily diffuse towards the microbial cell, cell, and once it's like close to the bacterial cell, they interact with the cell surface through the electrostatic interaction, and this electrostatic interaction occurs due to the presence of various charge on the or functionally charged functional groups on the carbon dots, and also the various oppositely charged particles, which are components which are present on the surface of the bacteria. And as this happens, the carbon dots, they form a physical barrier around the bacterial cell, and then it prevents the bacteria from spreading and also like getting access to the nutrients from the surrounding environment. As a result, this results into the physiological metabolism inhibition of disturbs the physiological metabolism, and this leads to stress and then death. Another one is the inhibition of biofilm formation. As I mentioned, said, carbon dots are very small and they can easily penetrate into the cell membrane of the bacteria. And once they go inside the bacteria cell, they disrupts the various signaling pathways that is necessary for the biofilm formation. As a result, the biofilm formation is prevented, and this biofilm is a very protective environment for the bacteria. So if it's not formed, the bacteria cannot be protected from the antibiotics. And then another one is when, as they are into the intracellular region of the bacteria, the carbon dots, they bind to the nucleic acids like DNA, RNA, and then they change their conformation. And because of this, the functions such as like replication, transcription, and protein synthesis, which are like vital, important functions for the bacterial survival and proliferation gets inhibited. Another one is in the presence of light. So in the presence of light, carbon dots, they act as photosensitizers. So carbon dots, they absorb light and then they produces reactive oxygen species, which act as antimicrobial effects. So when carbon dot receive rise, the electrons, this is in the ground state. So when they receive light, the electrons, they get excited and move to the excited singlet state. So now when they are in the excited state, the electrons can come to the ground state in two ways. One is either through the fluorescence by emitting light, or without emitting light via releasing the heat or energy. And then at the same time, some of the electrons, it can also go to a little bit lower state, or which is called triplet state. So the electrons from this state can also either come to the ground state via phosphorescence, which is similar to fluorescence, or via the release of heat without emitting light. So at this moment, electrons, what they do is they also undergo a photochemical process, two chemical processes. This is type 1 and type 2. So in type 1, what they do is these electrons, they transfer the energy into the surrounding molecules such as water and oxygen, and then this forms superoxide radicals. And then at the same time, some electrons, when they come back down to the ground state, they release single oxygen, singlet oxygens, which are very reactive. So this process keeps on continuing as long as there is light. So once the as oxygen reactive oxygen species and oxygen singlets are very reactive, and they causes oxidative stress to the bacterial cells. and then they destroy the cell wall, the cell components like proteins, DNA, and then causes cell leakage, and then this leads to the death of the bacteria. Polyalphalia longifolia. So mankind have been using medicinal plants since very ancient times, and medicinal plants have played a very important role in mankind despite so many scientific developments and all. So one such plant is Polyalpha longifolia. It is an evergreen tree which belongs to the family of Annonaceae, and it is found in abundance in subtropical and tropical regions such as Malaysia, India, Sri Lanka, Myanmar, etc. So since I am gonna I'll be working on the part of the leaf, so I'm gonna talk a little bit about the leaf. So the leaves of P. longifolia is they are lanceolate, and the length ranges from 15 to 20 centimeter. It is glossy, and then it has like the margins at the ends, and when it's young it is light green, and when it matures it's darker in color, and it also has a very pleasant fragrance, very like it's not that prominent when it's in a green, but when it's dry it is very noticeable. The leaves of P. longifolia contains various compounds such as flavonoids, alkaloids, diterpenes, tannins, etc. And these plants or different parts of this plant were traditionally used to treat various diseases like skin disease, diabetes, body pain, fever, etc. There are various therapeutic properties of P. longifolia, such as antioxidant, anti-inflammatory, anti-ulcer, antimicrobial, and etc. Literature review. Jera et al. They synthesized carbon dot from neem leaves using one-pot sandbath method. Their carbon dot exhibited concentration-dependent biocompatibility and antimicrobial properties against 4 bacteria and 2 fungal strains. Another study, which is a review paper, they discussed about the antimicrobial application of carbon dust, and they highlighted their low toxicity, how easy they were to synthesize, and biocompatibility, making a promising alternative to antibiotics. Another study evaluated the antimicrobial activity of carbon dust in combination with ciprofloxacin against Pseudomonas, and the results showed enhanced antimicrobial activity and inhibition of biofilm. So another study synthesized carbon dust from turmeric leaf via one-step hydrothermal process, and the carbon dot also demonstrated photostability and effective antimicrobial properties against both gram-negative and gram-positive bacteria. Problem of statement. Over-reliance on antibiotics has caused great concern in the microbial infection, which is caused by various pathogens such as bacteria, fungi, viruses, and parasites. A study which was published in 2015, conducted by CDC, they found that global antibiotic consumption increased by 65% from 2000 to 2015 in those 15 years. And another one is rapid evolution of resistance. Because of the misuse and unprescribed use of antibiotics, bacteria or the pathogens have been evolving rapidly. They either mutate or they gain access to the resistant genes from their surrounding. A study conducted in 2019 reported that 4.9 million deaths occur annually, and if because of these antimicrobial resistant pathogens, and if nothing is done about this, then by 2050 about 10 million deaths is estimated to occur. So another problem is the increased cost and technical challenges. The development of new antibiotic is very expensive and technically challenging. The lengthy processes or pathways to approval to get approvals, which like takes about 10 to 15 years, is a major barrier. So all these issues necessitates the exploration of alternative methods to discover efficient antimicrobial alternatives to antimicrobial agents. So the objective of this proposal is, the primary objective is to synthesize carbon dots using P. longifolia leaves for an in vitro antibacterial investigation. And the specific objective is to synthesize carbon dots from P. longifolia leaves to characterize the synthesized carbon dots and also to assess its antibacterial activity. So the hypothesis for this research is that the synthesis of carbon dots from P. longifolia leaves will result in efficient and reproducible methods for producing antibacterial nanoparticles, and the characterization will reveal distinct structural and chemical properties which will be suitable for antibacterial application, and it will exhibit significant antibacterial activity against targeted bacterial strain in this in vitro study. So this is my methodology overall flowchart. I'm gonna talk in detail in the other slides. So my methodologies are based on the specific objectives. So the first specific objective is to synthesize carbon dots from P. longifolia. For this, mature and healthy leaves will be collected from the USM and then authenticated at the herbarium of School of Biological Science. Then the leaves will be cleaned with deionized water and then dried for about a week, and then cut into small pieces and then ground it into small powders using electronic grinder. And to synthesize carbon dot, once the small pieces have been obtained, it will be introduced into the furnace at 300 degrees Celsius for 2 hours, which will give brownish black residue, which will be further grounded into fine powder with the help of mortar. And then 10 milligram will be dissolved in 100 ml deionized water and then sonicate for a homogeneous and stable solution, and then centrifuge, and then filter using a syringe filter. And then the filtrate will be sent for dry freeze. After dry freezing, carbon dots will be obtained in a powder form. And then it will be stored at 4 degrees Celsius for further utilization. Specific objective 2, to characterize the synthesized carbon dots. So characterization involves various techniques to analyze physical, chemical, and optical properties of carbon dots. It is important so that the carbon dot meets the desired specification for the designated application. So TEM, it is one of the technique which is used to characterize carbon dots. It determines the size distribution and the morphology of carbon dots. For this, I'll be using JS Libra 120 and will operate at 80 kilovolt. So for this, a drop of carbon dot solution will be deposited onto a carbon coated copper grid and then dry at room temperature. Then the image will be like captured at various magnifications to visualize carbon dots. Then the size will be measured in different directions, such as longitudinally, transversely, and diagonally. Then the average will be calculated and the data will be presented as either mean deviation. Zeta potential, it is also another technique which is used to characterize carbon dots. It is used to analyze the stability of carbon dots or the surface charge. The surface charge will be assessed through or using dynamic light scattering device or analysis. It is a technique which is used to determine the distribution of small particles by analyzing changes in the light scattered from a suspension due to the Brownian motion. So that the data will be performed using ZetaSizer family software version 7.11 and Zeta potential which exceed plus 30 millivolt or less than 30 millivolt are considered good characters. UV-Vis spectra analysis, it is used to determine the optical properties of carbon dots. For this, UV-Vis spectrophotometer model UV3600 will be used, and LS solution UV-Vis software will be used. For this technique, 3 ml of 10 milligram of per mil of carbon dot solution will be transferred to cuvette, and the cuvette will be inserted into spectrophotometer's cuvette holder, and then the sample will be run in a wavelength range of 200 nanometer to 850 nanometer, because carbon dots, they usually observe light around 200 nanometer. Then the observance will be like observed using the LS solution by software, this software. Fourier transfer infrared or FTIR spectroscopy, it is also another characterizing tool. It is used to identify the functional group present on the surface of carbon dots. For this, I'll be using FTIR spectrophotometer. Then the data will be acquired using Perkin-Elmer Spotlight 400 imaging system. In this technique, about 1 milligram of my carbon dot will be mixed with potassium bromide at the ratio of 1:100, and then will be pelleted, which will then be positioned into FTIR sample holder, and then run the sample at a spectral range of 400 to 4000 per centimeter. Then the peaks in the FTIR spectrum will correspond to the specific vibration modes of functional groups. So the different peaks will denote different functional groups. Specific objective 3, which is to assess antimicrobial activity of carbon dots. For this, first we have to prepare the bacterial culture. The bacteria will be first cultured in Loria broth and then incubated at 37 degrees Celsius for 24 hours in an staking incubator. And then the bacteria, it will be harvested during its exponential phase via centrifuge. The supernatant will be discarded and the pellet will be dissolved in PBS, phosphate buffer saline. Then the concentration of the suspension will be measured, or its optical density will be measured at 600 nanometer. This is assessing the antimicrobial activity of carbon dots. For this, the previously prepared carbon dot, the bacterial solution will be adjusted to the OD of 0.1, and then the suspension will be diluted with the PBS that contains different concentrations of carbon dots, and then it will be seeded into 96 well plate and then incubated in a Staker incubator at 37 degrees Celsius for 24 hours, and then its OD will be measured again. So at the same time, the viability of the bacterial cell will also be calculated using this formula. This is after the treatment with carbon dots, and this is before the treatment with carbon dots. So scanning electron microscopy, this will also be done. This will be done to explore the impact of carbon dots on bacterial cells. For this, the carbon dot treated cells will be centrifuged. It will be like harvested at their exponential phase, and then centrifuged, and then washed with PBS thrice. And then it will be fixed with 2.5% glutaraldehyde at 40 degrees Celsius for 6 hours. And then the glutaraldehyde will be removed. And then it will be dehydrated using ethanol series of like different concentrations, such as 70, 90, 95, and 100% to like preserve the structure or the morphology of the bacteria, and also to the 100% ethanol is to make sure that the water is completely removed, and then it will be dry freeze again to make sure that its morphology is preserved, and then it will be subjected to scanning electron microscopy. So the expected outcome for this research is that carbon dots from P. longifolia leaves will result in efficient and reproducible methods for producing antimicrobial nanoparticles or carbon dots. And the characterization will reveal distinct structure and chemical properties suitable for antibacterial application, and will also exhibit significant antibacterial activity against targeted bacteria. So the conclusion is this study aims to synthesize green carbon dots from medicinal plants and evaluate the antimicrobial efficacy against pathogenic microbe. It is anticipated that green carbon dots will exhibit significant antimicrobial activity, demonstrating their potential in combating infections. Also, the eco-friendly synthesis and use of medicinal plants are expected to align with substantial principles, like highlighting an innovative approach to antimicrobial agent development. This is my Gantt chart and references. Thank you.
Thank you, Tapa. Okay, maybe Dr. Shahida, you could start with your questions or comments.
Thank you, Dr. Ong. Yeah, and thank you, Tapa, for your presentation. It's a nice work indeed. Your proposal and your slide. I have question about the. Do you have any positive control when you do the analysis that you compare with?
Oh yeah, sorry, I forgot about that.
Yeah.
Yeah. So in the antimicrobial assessment, there will be a positive and a negative control. Positive control will be the bacterial growth itself, because we have to see the turbidity, like how well the carbon dot is works. And then for negative control, the media or the PBS.
Okay. When you do the characterization study, or maybe you can show us the slide where you produce the carbon dots from the leaf. Okay, next slide. Okay, here. Okay, when you extract the carbon dots from the, after you sonicate, how you know it is the carbon dot? Any validation here?
Oh yeah, for to make sure that it's the carbon dot is synthesized, I will be take a small portion of the solution and then take UV of 36500. I forgot that. Just yeah, and then go to the dark room and then go to the dark room and then flash the UV light. And if it gives like bright color, like any blue color, green color, or any orange, red color, then it means there is carbon dot.
Do you have any positive control here? Like you already, can you buy carbon dots in the market? Is it commercialized?
According to me, I mean, to my understanding so far, so this is green green carbon base, so which means this is from plants, but other carbon dots which are not synthesized from green plants, there are other carbon dots which are synthesized by doping, like using chemicals. I think those are in the market, but green carbon dots, I don't think so it's there yet.
Because my concern is, if let's say you take the carbon dot solution and bring to dark room, and if let's say the intensity is low, so you're not sure whether this is a carbon dot or because of low concentration of carbon dot. If you have the positive here, maybe you can compare. If let's say the intensity is equivalent to the positive control, then you know there's a carbon dot inside your solution. That one I'm not sure, but you can ask your supervisor to discuss about the consistency. Have you ever measured in how many solution of your carbon dot solution, what is the concentration? Is there any, how to say, do you know, do you have you standardized the concentration from how many grams of leaf, then how many concentration of carbon dots that you obtain?
That I haven't done yet.
Because I can see from your method, I'm not sure whether I didn't understand or not. I didn't see any consistency. Like if you standardize the concentration, let's say you extract carbon dots from 5 grams of leaf, then it brings you how many concentration, and then from this concentration, you consistently use it to analyze for every batches. You know, you're not going to analyze all those analysis in one day. You're going to do in multiple times, right? So my concern is batch variation. If you have standardized concentration prior to this analysis, I think that would be better. Can you go to mechanism of action? I'm not sure whether I read in some parts of your proposal, you mentioned about the future work is to study on mechanism of action. So what do you mean by that?
So, like I mentioned earlier, the mechanism of how carbon dots work against the microbial is not yet, it's not confirmed yet. People, researchers, they are still working on it, so they don't really know how it's doing its work. So maybe in the future, I could go and explore more into this field.
So the future plan is not part of your project. Is something that you propose in future.
Yeah, I don't think so. I can do it. I mean, with this time frame, I don't think so. I can, continue my further future work.
Understand.
So,
because I'm just concerned because I read from your proposal you put it in future work, so I'm not sure whether it is part of your work or you plan for. I think the one that you should put in your proposal is something that you within your project, within your responsibility for the project. My final question is, now you say that these carbon dots could be a substitute to antibiotics. So in what form it can be delivered to patients? Right now, we receive antibiotics in the form of drugs, like tablet. So your proposed carbon dots, in what form it can be delivered to the patient?
So this is like at the moment we are like, nobody has reached at that level of research, but I think if you wanna treat the wounds or injuries which is on the outer side, you can use it as a like solution. You can apply and then you can like expose it to the light, and then it will be treated. Obviously it cannot be in the lotion form, so in a solution form, and then if we have to inject it into our body, and then recently we visited the IPPT, the nuclear therapy place. I forgot the name. So what they do is like they were using this radioactive material, and then like obviously it also it's. I thought like this is somewhat similar to that. So I was thinking. So what they were doing is that they activate the material that they're gonna use for the therapeutic, and then they just prepare it now and then immediately they transfer it to the patient chamber through some passageway. So I was thinking maybe for this also, I mean, because it's an electron and it's not gonna stay in the excited state for a long period of time. So we cannot keep it for a longer period, right? So I was thinking maybe we could do something similar to what they were doing in the hospital.
And from your reading, what are the stage that people are researching with carbon dot?
Sorry, can you repeat the question, please?
From your reading, what you know about the carbon dots that people have produced, what stage are they now?
So at the moment, what people are what researchers are struggling is that like there is no any standardized procedure to synthesize carbon dots. So everyone is like using different methods to synthesize the carbon dots, and they are obtaining different carbon dots, different features, featured carbon dots. So there's no any standardized. So at the moment, they're just trying to figure out the one standard, one good method to synthesize carbon dots.
Okay, good. Okay, Dr. Ong. That's all my question.
Yes, thank you, Dr. Shahida. Then we pass to Dr. Chiu now. Dr. Chiu.
Yeah, thank you, Dr. Ong. And hi, Pushpa.
Hello, doctor.
Yeah, I have a few questions for you. It was written in your proposal that there's one part on zeta potential. It was written that carbon dots with zeta potential more than positive 30 millivolt, right? Or less than negative 30 millivolts are considered stable. Then the zeta potential must be maintained within this range to ensure the stability of the CDs. So I'm a bit confused here. I just would like you to maybe explain, because I'm not familiar with zeta potential. What do you mean by this range? Because one is more than 30 or less than negative 30. Then within this range means what?
Maybe there was typo. Okay, so zeta potential, it is first of all done to find the surface charge of the carbon dots. So if the surface charge is positive 30 volt, more than that, or if it's less than 30, so more than 30 or less than 30 is
30.
considered good.
Yeah, but the sentence after that is that the zeta potential must be maintained within this range to ensure the stability, meaning it's above positive 30 and below negative 30. It's not within that positive 30 and minus 30, right?
Now I'm confused.
Yeah, because the written text is a bit confusing. That's why I just want to clarify from you, understand a bit from you, whether the range means within the positive 30 and negative 30 or above positive 30 and below 30 negative. So
within
the positive 30 and negative 30
is 30.
Is 30 is stable, right?
Yes.
So I think you
between
should
positive 30 and okay.
negative 30, right? Because it's contradictory, the two of them. The two sentences contradict themselves, okay?
Thank you, doctor.
Okay. Yeah, in your slides, you mentioned that FDIR is used to assess the antimicrobial activity of your carbon dots, right? FDIR in the slides. It's under antimicrobial activity to assess.
Oh, it's a mistake.
Pardon?
It's my mistake. Sorry.
Oh, so it's not to assess antimicrobial, is it? It's to characterize.
Yeah, it's supposed to be characterized.
Okay. And another question is why you use only gram-positive Bacillus subtilis for your work? Only one bacteria in your work.
At the moment, that is the available one that I have.
You mean
But
you don't have any other bacteria?
I'm working on it.
Yeah, because we have only one and the money constraint, so we propose what we have.
Oh, so you only want no gram negative or any other?
Yeah.
Okay, my concern is that there's only one bacteria, so this is for your reference lah. Only one
Okay.
bacteria. Then another one is your objective number one is to synthesize the carbon dots, right? Then the outcome is you stated that your outcome for objective number one is to find an efficient and reproducible preparation methods, right? Your objective number one is to synthesize carbon dots. The outcome is to find an efficient and reproducible methods. So how do you correlate your outcome to your objective? How do you ensure that your methods is efficient and reproducible, whereby you do not like quantify the yield or something along that line?
Yeah, that was my mistake. I wasn't quite aware of that, so
Yeah,
I'm not sure if I can fix that.
because you understand my question, right? Because you
Yes.
have only one fixed method, right? 300 degrees Celsius for 2 hours, right?
Yes.
In the furnace, then after that you filter and all that. Then how do you ensure that there is an efficient and reproducible methods, whether it's the most efficient or unless you determine the yield or something like that, right? You quantify or something to justify your outcome.
For this, I might be doing the quantum yield.
Then why do you fix the 300 degrees Celsius for 2 hours? Because in your written proposal, there's a table 1.1, right? There are many others temperatures and duration of for the synthesis. So why do you fix at 300 degrees Celsius for 2 hours? Any particular reasons?
Like it's said earlier, like there is no any specific condition, for to synthesize carbon dots. So our aim is to try to maybe we could invent some easy way to synthesize carbon dots. Like they haven't done this. We are using the furnace, and then I don't think so they have used the furnace. So we're just trying to find a new way to synthesize carbon dot. So there's no any specific. It's just
Okay.
just trying to explore more.
Yeah. Okay. I think maybe the title to me is not very specific. So maybe at the end of your work, maybe you can have a look at it because it's quite general compared to your work. I mean your research title lah. I think that one will be discussed later at the end of your work. And my comment for your written proposal, there are quite some errors on like the use of C dots, CDs, CDS, QDS, Ds. Different for C. Actually, your carbon dots, the abbreviation you use is CDS, right?
Yes.
Then I can see that C dots was used somewhere. QDS on page 7, D on page 14. So all that I think you have to go through and do some minor correction on your written proposal as well. Some like the format of writing scientific names, and spacing and all that minor mistakes. So maybe it'd be good if you can have a look at that. Okay. I think
Thank you, doctor. Thank you.
yeah, I think that's all from me. Pushpa?
Thank you, doctor.
Okay, thank you, Dr. Chiu. Papa, you just take note with that. Maybe I can put in one point is if you need other gram positive or gram negative bacteria, if you need it, discuss with your supervisor. If you need it, then I have some. You may choose from our stock. All right, if you need it.
Thank you, doctor. Thank you.
Thank you, Papa. So,
Thank you,
finish.
doctor.
I pass it back to Dr. Sasi.
Yeah, thank you, Dr. Wong. So
Thank you. Okay.
now we move on to next presenter, Ms. Yap Zijian, under the supervision of Dr. Eugene. And the title of proposal is Unraveling Environmental Microbial Diversity, Metagenomic and Bioinformatic Exploration. And the evaluator is Dr. Fatma and Dr. Nick Hughes. So,
wait, doctor, did you get the wrong, not the wrong, sorry, the older title?
never mind, you can project and you can tell your new title. You
Oh,
can
yeah,
start.
okay.
Yeah. You
All
can
right, sure, no problem.
Right. So you can just proceed with your new title.
Okay. Good afternoon. My supervisors, Dr. Eugene, and others.
Yeah, good afternoon, Leo, and also
Yeah.
Dr. Sasi. We have to wait for a little while for Dr. Nick. I think she hasn't joined yet.
Okay, Dr. Nick, yeah.
Dr. Nick hasn't joined. Yeah, I've sent her the link already.
Okay, so just wait for a while.
Okay.
Okay, Dr. Nick Hughes is here already.
Okay, so can proceed.
Yes, Dr. Dr. Nick Hughes, Dr. Fatma also here. Leo.
So today I'm going to present my proposal, which is a title related about machine learning based prediction method in identifying skin cancer type by their microRNA expression profile. So this is prepared by me, Yap Zijian. Okay, you can call me Leo also. My matrix number is 222024449. And then main supervisor is Dr. Eugene, and co-supervisor will be Dr. James and Dr. Lee from Codon Genomics. Oh yeah, because I got this project from the external company. So yeah, they will be my co-supervisors in helping me to doing the more technical part about machine learning. Okay, so how can I here? So introduction. Skin cancer. Skin cancer characterized by the abnormal growth of skin cells, primarily arises due to exposure to UV ray from the sun. Okay, this exposure induces DNA mutation, triggering uncontrolled proliferation of skin cells within the epidermis and form tumors. Right. So, skin cancer. Skin cancer, it can be categorized into 3 types. But if you want to put it into like 2 groups, you can put it into melanoma and non-melanoma. So melanoma is a kind of skin cancer that starts in the melanocytes. So melanocytes are cells that make the pigment that gives skin its color, and the pigment is called melanin. While the non-melanoma skin cancer refers to all the type of skin cancer that occurs in the skin, they are non-melanoma. Very straightforward. So the very common type that the cells might be becoming cancer is these squamous cells and basal cells. So we call them squamous carcinoma and basal cell carcinoma. Where's my... Yeah, so this is a typical picture of melanoma. We can see a black spot here, and this is pictures of non-melanoma cancer, skin cancer. Okay, so a conventional diagnosis of skin cancer is via visual inspection or biopsies. So visual inspection, typically the dermatologist often will begin by visually inspecting the skin with, I believe now the technology used for visual inspection is called dermascope. Okay, and then other than that, also they will make biopsies. So if a suspicious lesion is found, a biopsy may be performed. And there are different types of biopsies, including shave, punch, and excisional biopsies. So why do I want to talk about this is because there are several challenges in skin cancer diagnosis. First, you have visual similarity. The different types of skin cancer can resemble benign lesion on each other. Okay, making accurate visual diagnosis is hard, okay, even for experienced doctors. And then overlapping features, some features associated with skin cancer, such as irregular borders or color variation, can also be present in benign lesions and other skin conditions, leading to misdiagnosis. And variabilities, skin lesion can exhibit a wide range of appearance, making it difficult to establish a consistent diagnostic criteria. And last but not least is very subjective. So visual diagnosis relies on the experience of the dermatologist. So this may introduce variability and inconsistency in the diagnostic accuracies. So why I want to brought this is because I want to I want to solve this by using a machine learning method approach. So conventional method of skin cancer diagnosis often rely on visual inspections and examinations. So we can be subjective, time-consuming, and very prone to error. Okay. Therefore, in these studies, maybe I should make this a study. Sorry. Propose to use microRNA as a key biomarkers with the aid of machine learning. Okay, together with the public data that is already well established on internet in enhancing skin cancer diagnosis. So thereby, the main aim of this study is to explore the possibility of using machine learning as a prediction tools in identifying skin cancer types. So the objective here is to explore various machine learning as a prediction tool in diagnostic skin cancer type based on microRNA expression profile. So the specific objective will be, first, what I want to achieve from here is to, of course, when we got the data, we need to select what is the proper appropriate data to use. So objective one will be to select and reduce dimensionality of microRNA expression profile data, because this data is come from public data set. To select candidate mRNA expression profile as a biomarker in the 3 major skin cancer types. So we'll be focusing on melanoma, BSC, and SCC first. And then to develop, second objective is to develop, so based on this data, the second objective is to develop the predictive models. Okay, these are the models I propose to use by training a data sets from the, Miss wrote here. Okay. So literature review. Okay. So microRNA are short non-coding single-stranded RNA molecule, typically 20 to 24 nucleotide long, that regulate gene expression at the post-transcriptional level by binding to mRNA. So they play a crucial role in the oncogenic activities by managing the mRNA expression during the cell cycles event. Okay. And so these are the online literatures, online papers that has been found. The microRNA, there's the role of microRNA changes in targeting the protein and causing the cancer cells. Right. So other thing is machine learning. machine learning is a method to train computers by developing algorithms to serve the purpose of identifying patterns and making predictions with minimal external intervention based on the data and statistical model presented. So the reason I highlighted the supervised learning here is because in my studies, I'll be mainly focusing on supervised learning because we are dealing with more well-labeled data. It's not unlabeled data. So these are some examples of papers that has been proposed to use the machine learning in medical studies, particularly in cancers. So we can see one provide overview of machine learning used in a cancer prediction and prognosis, and then explore the use of artificial intelligence and machine learning in cancer imaging. And last but not least, something related to skin cancer is Ahmad in 2020, they discussed the various machine learning and deep learning techniques used for the classification of skin cancer through image processing. So my method, so the general workflow of this method is first we'll start with data mining. Total of 452 skin cancer transcriptomic files from 448 cases are going to be extracted from the GDC data portal. And then after that, we'll do data cleaning. After we got the data, and then we need to analyze to identify the relevant microRNA expression data sets that are associated with a different type of skin cancer. And this is going to be performed by using Python with Scikit and R. And then third is data modeling. We will be, based on the data, we will train 5 predictive models from SVN, DTS, random forest, and linear regressions. Okay. And then the second things we do data validations. So the trained models were going to be evaluated under 4 metrics. And then of course, the microRNA data set that will be validated using the online web server called GDAC. Okay. So we were comparing with this to validate the data. So the expected outcome is, of course, answering to the objective. The feature selection techniques will effectively identify a subset of microRNA expression data. And then the second thing is related to model performance. So after we got the predictive model, we will train them, and then hopefully we will identify the best model with superior performance, okay, based on the metrics we'll be using proposed in the methodology. So summary, to this stage, I'm still at proposal writing. And then due to the limited time frame of this project, the study is mainly focusing on mRNA expression profile of skin cancer tumor. So in the future, as I talked with the company also, they say that if we're able to complete these very fundamental things at a very short time frame, maybe we could proceed with the downstream process But I'm still not sure yet and what to do on the downstream process. Yeah, so basically this is my Gantt chart. And then that's all. This is the reference. So I will end my presentation and move to Q&A session.
Yeah, thank you, Leo. So now I open for Q&A. So maybe Dr. Fatma, you have any question or suggestion?
Thank you, Dr. Sasi. Thank you, Leo. I think maybe Dr. Nius dulu, boleh kah? But Dr. Nius, I think you are most familiar with this. And then I'll continue with you.
Okay, no problem. Actually, I'm not that expert too in machine learning, but I have a few questions and maybe comments. Okay, thank you, Leo, for the presentation. Thank you, Dr. Sasi.
Sorry, Dr. Nick, can you be louder a little bit?
Be louder. Okay, all right.
Yeah, raise the volume a little bit.
Okay, thank you, Leo, for the presentations. Okay, I just want to ask you regarding the data, right? So you are only focusing on the transcriptome data, or you also include the microarray data as well in your analysis?
I'll be only focusing the transcriptomic data.
Okay, okay. So it's not involving microarray data.
Yeah.
Okay, so when you validated, okay, you're saying that when you have the candidates for microRNA, right? So, and then you try to validate it using the online web server, right?
Yeah.
So, meaning that you just only targeted the microRNA that have been reported previously, right?
Yes, yes.
So, but I think you probably get a new candidates during your analysis.
Oh yeah, that is also written in my proposal. Also, that will be the future work lah. Because given the limited time frame, there will be. I'm sure there's some ethics thing you need to apply, and so if you want to test it, bring it on bring it onto a reality in doing this. So that will be taking a longer time lah. So that's why I didn't include this.
Okay, because I think it's if you have additional microRNA that could be potential for the future studies, could be new findings of the microRNA as a biomarkers, as despite just depend on what people already reported. Okay. Okay, so in machine learning, there are two approaches, right? Supervised and unsupervised. So you choose the supervised machine learning. So what are the difference between supervised, unsupervised, and why you choose the supervised approach for this study?
Okay, so the main difference between supervised learning and supervised learning, when we want to use that is that under a condition with whether you have a label data or not well labeled data. So what it means is label data is that this data you has already know is provided from like coding from this source, right? Unlabeled data is something that is you're not sure with. So since we already have dealing with the data from the public data sets, it's already told us that this cancer leads to this microRNA expression. So this is a very well labeled data already. So it's unnecessary to use the unsupervised learning here.
Okay. So there are quite many it's like 5 algorithm that you're going to use, right? So is it all these 5 are all I mean all the algorithm that we have for supervised learning? So you want to try all these supervised algorithms in your study?
Actually, there is some the what I propose is the 5 data, right? I'm just saying the 5 these predictive models, right? Am I getting your question right, Dr. Nick?
Yes. You want to use 5 algorithms. I just wonder whether it is doable for the time frames you have like 6 months, and I'm not sure whether it is doable to use all these 5 algorithms. Whether you have enough time to do that.
I think it is doable because when I go into the company and asking them, they said that usually in transcriptomic studies are the work that takes a lot of time, the very time consuming is at the very beginning where you have to process the very raw data. And then for modeling, actually, if you already have the data, it's actually not that time consuming, like they say. But I'm still not sure, but they have give me a image saying that, okay, I can complete this and I think I'm confident enough to do this.
Okay. perhaps I'm not sure. Perhaps maybe if you don't have too much time, you can choose one or two the algorithms. I'm not sure also what is the durations or the period of time that you're going to take for only one algorithm. Yeah. Yeah, I think that's all from me.
Hey, Dr. Fatma, any question?
Okay. Yep.
Thank you, Dr.
I'm here. Leo,
Yes.
you can hear me clearly, right?
Yes.
Okay. So I think it's interesting that your project, I think this is the first, is it the first project, machine learning, presented in a proposal level at Inform? I don't know.
I think so because in terms of machine learning, I think so at Inform level. Yeah.
Yeah, I think this is the first. Congratulations, Dr. Eugene, and also Leo, to be the pioneer for this kind of work.
That's exaggerating.
Okay, so first of all, my question, when you have this, at the end of the day, this, I assume that this is a essay. This kit or this whatever essay that you're going to develop. Your main aim is to what? To early diagnose skin cancer or to stratify the patient according to the subtype of skin cancer or what?
Okay, the main goal of this study is to increase the diagnostic accuracies of the skin cancer subtypes. Yeah, because as I mentioned before, the challenges in diagnosing for dermatologists to diagnose what type of skin cancer is this, it sometimes it can has a lot of factors affecting the diagnostic accuracy, like just now I mentioned. So to the studies now, what they are focusing is, and here they are using the machine learning also in aiding the visual inspection. But how accurate is that? I'm not sure. According to a report, I think I mentioned in the proposal is about how many? I forgot. 60, 80%, something, if I'm not mistaken. But it still will cause error lah. So, and the thing with skin cancer is that you have to diagnose it early. Early diagnosis is very, very, very important. So yeah, that's the rationale of why I raised this study, proposed this study.
So you're saying that even though early diagnosis is important, but your study is to do subtyping or to subtype patients according to the type according to the type of skin cancer.
Yeah, you can diagnose the skin cancer easily, but to put them into subtype is difficult. And if you are given a wrong result to a clinician, maybe they could provide wrong therapeutics, treatments for use, and maybe it can lead to ineffective treatments.
Okay, so I think maybe you could highlight that in your introduction. When this subtyping is very important because different subtype would have different regime therapeutically. Yeah. So you should mention that lah. Why? So I think every time when we develop essay, we need to highlight what is it for. Is it for early diagnosis when it comes to cancer lah? I don't know about infectious disease or other disease, but when it comes to cancer, we need to identify that do you want to do early diagnosis or you want to assist in therapy decision.
Okay.
Yeah. So in your case, you want to assist in therapy decision. So in doing that, you need to subtype the patient according to the subtype lah.
Yeah, but the aim is also to provide early diagnosis lah, because the survival rate survival rate for early diagnosis patient is relatively high.
Okay. So
Yeah.
coming to the early diagnosis, in your study, can you go to your methods?
Yep.
So when you are saying that you want to do early diagnosis, the methods, the one with the 3 point, the one with the 3, yeah, this one. So in this data mining, the total of 452 skin cancer and early diagnosis, usually you have a counter sample lah from normal.
Oh.
Okay. So usually that's the how do I say, that's common lah, because you need to have a reference and compare the patient. In your case here, you say for early diagnosis, but looking at the data that you show here, it's only coming from one group, which is from patients sahaja.
Oh.
So maybe, but this is at proposal level. I'm sure your understanding will expand a lot when you do this now. So bear in mind that lah, and do discuss with your supervisor. And that's why you really need to ask whether this is for early screening or is it for subtyping according to type, subtype, to assist in therapy decision. Because these two are a whole lot different design in experiment if you were to do it.
Oh, okay. Now I get it. What you mean is that early diagnosis is to determine whether the patients have cancer or not.
Yeah.
Is that what you're trying to address?
Okay. So my next question, when you put this 452 skin cancer from 458 cases, you already identify which data you want to use from the public database?
Yeah, this is already filtered. So
You already started lah.
Yeah, I already got the data, but I'm still yet to proceed to number 2 in doing data cleaning. We still need to identify, because there's some others microRNA that is expression profiles is not relevant to the skin cancer, so we still need to clean up all these.
I see. Okay. Okay, now the same question with Dr. Niu study lah. So you are not developing an algorithm. You are using or utilizing these 5 algorithm and testing your sample, right?
Wait. I think. Wait. Yeah, I think yes. Using these 5 machine learning to develop an algorithm. Actually, what I define algorithm is that the predictions tools that able to help you to.
So you're not developing the tools, the algorithm. You don't do the coding, whatnot. You are using the one that people have used. You just put your data in and see whether it can differentiate between the subtype, kan?
Yes.
Yeah. So that's what you do. So coming back to Dr. Niu's question, I think in the time, because you're not developing the algorithm, you are using the one that people have already developed, kan?
Yeah.
So there are 5 that you are testing. And my question, are you going to compare all these algorithm according to the parameter? You mentioned the parameters, kan? The parameters that you're going to get.
Yeah, after using we got the models from this. So we were.
Can I see the parameters that you're going to compare?
Here, the accuracy, F1 score, ROC curve, and confusion metrics.
So these are the parameters that you're going to extract out from the all 5 algorithm lah.
Yes.
And then you will come up with the best. You will show that, let's say, model number 5 is the best algorithm for this skin cancer that to answer the objective that you're going to do.
Yes.
Okay. Okay. I think my comment is this is a very good pioneer work involving machine learning at Inform. Congratulations. And I think that there's a bit here and there lah. My question regarding the what in the end, what do you want to use it for? Whether for early detection or to assist therapy decision? Because these two design of experiment is different. So I think for to answer whether that or that, the design of experiment, I think Dr. Wei Kang from Modern Genomic will help you lah, and also Dr. Eugene as well will help you to design it lah. But overall, yeah, I think it looks sound. Are you going to position here at Inform or at Cordon?
Sorry, me.
Yeah.
So in next semester, hopefully I will be in KL and doing in Cordon Genomics.
Good. So you will be placed dengan Cordon lah for the project.
Yeah, I'll be going to their company with their aid, so and then help me to doing
Good.
machine learning.
I think that's our motivation lah, for you to be exposed with the industry punya experience. So good to hear that you're going to be there. I think good luck, Leo. This is a good project, good pioneer project at Inform.
Alright.
Looking forward to see the progress.
Okay, sure.
Okay. Thank you.
Thank you very much, Dr. Farma.
Okay, thank you, Dr. Fatma and Dr. Nick Hughes. So, Leo, please take notes all the concerns raised by the evaluator. So, with that, I'd like to end this session. Thank you. Thank you, everybody.
All right. Thank you, Dr. Sasi.